Yes, hello. Sorry, just a second. So I guess everyone can see me. So this is me, Supantha Dey, student of genetic engineering and biotechnology at University of Dhaka. So I have currently been in a research fellowship program in Omics Logic Bioinformatics. Dr. Harpreet Kaur Madam has been my supervisor. So today I'm going to discuss my journey and how mentors and this internship helped me to gain knowledge and idea about developing a project and what I did. So what did I do actually? Well, first of all courses and projects I or you who are going to do this research fellowship will gain access to after enrolling. Well, they will guide you and they will be super helpful and they will be fun. I remember for the first time, I didn't feel bored when I studied and went through these courses all day. The knowledge you gain from these courses and projects will help you to do works for the research project later on. And also these projects will guide you in various courses as Dr. Ella already said, like in transcriptomics or genomics and in any, and you can also do the projects you will feel interested into. Also, apart from using bioinformatic tools, I was also exposed to the world of programming of data science and bioinformatics. It was a nice experience when you realize you can visualize your dataset with R and you can do lots of analysis. One thing that is not mentioned anywhere today, that is, you will know some cool people and make friends around when you do this project. For example, I did make some good friends who are undergraduate as me. I came to know people who are senior researchers or pursuing PhD. It is very helpful when you get this exposure. I think I talked enough about why I love it here. I'll give you a short summary of my work. So this is the research question I choose. So this is the one, how the third dose of malaria RTS-S1 vaccine is associated with controlled inflammation and increased protection. So before going further, I want to tell you how I came to this research question. Well, when I chose a research problem first, it was vast and not specific. I remember my mentors helped me to be specific and they guided me like you have to choose a good data set. They have to be properly utilized, and also the problem that has to be unique and novel. The first time I choose a problem that was a novel and the data set was so small, and they all the processes. Okay, so I wanted to mention that. Also, 4 or 5 months ago, I didn't know much about bioinformatic analysis or how to deal with dataset or how to interpret result or how to do multiple type of omics analysis. So here in the internship, you will learn all of those from the courses and projects. And not to mention, the mentors will guide you whenever you are stuck or you do a mistake. So I want to tell you why I choose malaria, because I guess Anita just before me, she explained everything why it is still a crucial problem and it is taking lots of lives. Those of us who live in Asia or in Europe, we do not understand that, but in Africa it is affecting very badly. So I'll now show you some of the analysis that we did, and I'll not go into the technical details. In this slide, you can find out that after differential gene expression analysis in AGR, we found 219 genes are significantly expressed. While we compared between pre-vaccination state and the day of challenge. The day of challenge was 3 weeks after the vaccination, where controlled human malaria infection was applied. Also, you can see the threshold for our result. Also, here you can see it is a volcano plot for all the genes. We use PSEC to achieve this, and this is the heat map. I also want to mention that you can all do it in the platform that Dr. Ella showed you before on, and also you can do it by yourself in the R if you go through the courses. Also, this PCA plot here, for example, let us see why this is important. So you can see these samples, they're the below one, the day 40 samples, they're different from other groups. They're not together. Also, day 1 and day 3 here, these two, I guess you can see, sorry, here, yeah, day 1 and day 3 samples, they are together from other groups, and the day of challenge here, they are here. So we can interpret many things. For example, in our study, we found out that cytokine-related genes were significantly expressed. So what the analysis this conclusion, overexpression of cytokines is associated with driving forces of death in malaria related disease. So here we try to find out how those genes which are significantly expressed affect the pathways, and we did enricher, as you can see, and in the platform we did the human gauge analysis. Here we can see the significant genes in our analysis. They are involved in cytokine related pathway. So we came to this conclusion, how malaria vaccination helps to downregulate those genes, the cytokine ones, which would otherwise, increase the severity of malaria and increase the chance of death. So we visualized our data set and we analyzed it, and that is all. So I will I haven't gone into the details because there is short time, and I think it would be best if I give you an idea what I did. So that will be all. And I want to thank Pine Biotech for giving me this chance to present and do this analysis. And thank you, everyone. Thank you.